Are you ready? Who would like to go first? Caroline or Lena?
I can go.
Oh,
Whichever.
Okay, you both don't care. So Caroline, I think you, well, I don't know. It was a jinx, so I'll let one of you. Caroline, you go. Is that okay?
Okay. So my article that I found, it's actually very recent kind of thing, but it's about these three students at Vanderbilt. They're seniors, and they made a phone case that is resistant to pathogens. The article was called they made a self-sanitizing cover phone case, but it's a little bit more than that. So after two of them became infected with COVID-19, the three wanted to turn their attention to ways they could help in the fight against the virus, and so they came up with this idea, and one of them found that copper is a really good choice of material for this because copper begins killing pathogens on contact. According to the New England Journal of Medicine, not even one viable microbe is detected on the copper surfaces after 4 hours, which is much less time than like the multiple days that it takes for the virus to die on things such as stainless steel or plastic. So these three guys launched a company named Aries to sell their phone case, and they're donating one phone case to a COVID-19 frontline worker for each one that is sold. I looked up a little bit about copper's effects. So copper is effective at killing pathogens because of its ionic nature. It is positively charged, which allows it to bind to the negatively charged cell walls of pathogens, and it disrupts their cell wall, allowing for the leakage of cellular material out of the cell and also like contaminants into the cell. And it also destroys the DNA and RNA inside the cell. And this is not really a new finding because this property in copper has been used for like a long time, or like it's been known for a long time. Like centuries ago, people used to know that if you drink water from a copper cup, you were less likely to get sick. And so, but these properties are still not widely used in places that would benefit greatly from it. So like replacing certain equipment and furniture in hospitals with copper variations would have an enormous impact on the number of people who get healthcare associated infections when they stay in hospitals. So just things like doorknobs and like bed rails, that would really make an impact on that. And so oh yes, and then the how I thought this impacts us as Christians is like these three guys who came up with this company are a really good example of how we as Christians can think about having an impact during this pandemic, because they were thinking about like ways that they could use their talents and gifts to like help those on the front lines and those elsewhere. So I just think that's a really good example of how we can be looking around for things that we can do. I mean, small things or big things, just ways we can help in the cause for this crisis. And yeah, that's my report.
Cool. Does anyone have a question for Caroline? I love it if there's at least one question.
I have a question. I don't know if you know the answer to this, but is the reason they're not using copper because it's more expensive for like the doorknobs and okay.
Yeah, I did read some about like that it is more expensive, and like just replacing the stuff they already have would be really expensive as well. A lot of it's like stainless steel right now in hospitals, so it would be pretty expensive.
This is not necessarily related to microbiology, but related to your presentation. Do you happen to look up how much those phone cases cost?
Since they're donating like one for one, like one to the front line, they're like, I think they're like $20-something each.
Okay.
but it's not too bad actually.
No, that's not outrageous. I thought you were gonna say like $75 a piece and be like, huh, okay, cool. That's interesting. I like that. I'd never heard about that for copper. So my takeaway is if you have a penny, you should be sucking on it like candy, right? But don't swallow. Is that right, Caroline? Do you think that'll help?
Yeah.
That would be interesting. I don't know. Okay, cool. So copper. All right, that was a really interesting article. All right, and kind of cool that it's just a few hours west of here. Where they're working on that. Awesome. All right, Elena.
I did my presentation on, or the article that I found was about the influence of soil on the immune health. So basically what it was is researchers started saying that along the border of Finland and Russia, that allergies were more prevalent on Finland's side than on Russian side, and they wanted to know what that was or why the cause of that was. And what they did is they predicted that differences in allergies was due to the soil, just because a lot of Finland citizens live more urban lifestyles than Russians, where they live more rural. And so basically what they did is that to prove that soil was the cause for the differences in allergies, they placed some mice on clean bedding and then had another grouping of mice in a cage in a stable with like other animals and sheep, and then they sprinkled different types of soil in the mice's cages. So after 6 weeks, the scientists saw that there was like significant differences in the lungs and then of different just bacteria in the gut biomes of the mice. They saw that the mice on clean bedding were more susceptible to lung inflammation than the mice who had been in the soil environment. As well, they observed that the gut biomes of mice who had lived in the soil had more bacteria like Bacteroidetes in the Bacteroidetes phylum than the Firmicutes phylum. And basically what that is Bacteroidetes are good for your body. They produce favorable metabolites and they reduce inflammation. Whereas Firmicutes are known as bad guys, and increased number of Firmicutes have been correlated with type 2 diabetes. So that was a pretty interesting finding. Mm-hmm. And then basically after the experiment, they just concluded that spending time in nature with soil provides a wide-ranging amount of health benefits, and that it was partly the soil that was playing an influence in the differences in allergies between the two countries. And then I kind of just said this was important to the field of microbiology because a lot of people are faced with allergies every single year, and that discoveries like benefits of different soils and their impact on reducing allergies, I think is important to healthcare workers for the prevention and treatment of allergies, as well as it brings awareness to being outside versus staying inside. And then that overall this information can lead to building immune systems in people. And then I thought that this information affected us as Christians because God calls us to take care of his creation and to spend time in his creation as he intended. And like science proves that there is benefits for being outside and being in his creation. So I just thought that was interesting. Mm-hmm, mm-hmm. And then that's pretty much it.
Okay, cool. Does anyone have a question? Any questions for Elena? Any questions? One question? I love awkward silence, even on Zoom. Okay, I'll start it off, but then someone else has to ask another question. So the soil, are they just doing soil and they're just saying there's nothing about the soil in the two countries. It's just that they're saying people in Russia are outside working in the soil more. So it's not so it's not like if they took soil from Finland and sprinkled it on the mice versus soil from Russia and sprinkled it on the mice, right?
No, it was just the amount of exposure, I guess, to the different types of soil and the microbes in the soil was building.
So it wasn't a soil specific to the country. It was just a lifestyle. Okay, I got you. Mm-hmm. Anybody else? Should I keep on asking questions till you think of one? Do you want to do that to your poor classmate? because this is a topic I love. I can go all day, and then you guys will be responsible for just looking over the PowerPoints on your own. All right, if that's the way we want to play it, question number 2. Did you happen to look up, so I think, and I can't see your paper, but I think the one group you're talking about, so you talked about firmicutes and you talked about Bacteroides, I think is the other one that you were trying to say, possibly, which is fine. Did you look up, is that like a specific class, order, genus, species? Did you happen to look that up?
In the article it says a phylum.
Okay, it's a phylum. I totally agree with you. So this is what's really interesting about the microbiome field, is that in every paper, and I had one paper that I published as a first author, and then I was on a couple other papers that had some of this information in it. And so every paper wants the phylum level reported. What do you think? What can you speculate? So they said firmicutes bad, Bacteroides good, right? Do you think there's a problem with that statement, potentially? If you're think about the phylum level.
I think, I know that you have both in your system. I think it's maybe the levels of how much you have.
Okay, so yes, I totally agree that the levels matter. But, okay, let's just say, what happens if I said you have 2 phylums, Christians and Muslims? Would you feel comfortable with a blanket statement that all Muslims are bad?
No.
You wouldn't, right? And I totally agree. And so this is the problem with phylums, all right? And you could say, would you feel comfortable with the statement that all Christians are good? And I would say probably not comfortable with that statement either. Because within each group, all right, you can make a generalization if you want. And in some cases, there's truth, right? But within that group, right, if I just say like Christian, Muslim, well, there's a lot of individuals that are slightly different. Some of them are good, some of them are bad in both groups. And so the thing that I love, well, that I don't love, it's just interesting, is that we rely really heavily on this phylum reporting. But there's plenty of research that shows that there are certain firmicutes that actually can be beneficial if you get really specific. And so it's a very interesting thing on the field. But I'm glad that you looked at the phylum. And so that's the problem, right? Phylum is kind of up here. Species and genus is kind of down here. And so, it kind of maybe gives you not the right impression. But for whatever reason, every microbiome paper, if you were to leave that out, they would ask for it. So, all right. So we still need another question. I'm getting the hard ones out of the way.
I have a question. Why did they choose like Finland and Russia? Are they close geographically? Like why did they choose those 2 countries?
It was like along the border of the 2 countries. So it was kind of interesting, I think, for them to see how they were close to each other, but they still had such different like types of allergies and just immunity to the allergies. So
Okay, so I'll take it. Before we move on, this is for the general group, not just Elena, right? So you don't have to necessarily leave her hanging. So this project is actually formed off of a hypothesis that was developed in the late 1980s. Does anyone know what that hypothesis is called? All right, basically that dirty things are good and clean things can actually be bad. Has anyone heard of that? Does anyone know what that is called? There's actually a really similar study. Interestingly, right, they didn't understand exactly why it was happening, but in the late 80s, right? Have you heard of the Soviet Union? Right, so what came down in the late 80s that was a symbolic end to the Soviet Union? Does anyone know? Any of my history buffs? It was in Germany.
The Berlin Wall.
The Berlin Wall, right? And so what was interesting is that Berlin is actually in East Germany, and so which was the communist part, but there was a West Berlin and an East Berlin, and the West Berlin was actually democratic, right? And so if you look at a map, it's almost like it was like this little island in the middle of the communist country. And so when the Berlin Wall fell down, what researchers noticed is that the West Berlin, which is more like we live, right, kind of a higher, you would say a higher quality of life. They had more allergies and autoimmune diseases than the people that lived in East Germany, right? They were actually, from an autoimmune disease allergy standpoint, they were healthier, and living conditions were a little harder, were not quite as sanitary, all right? And so that is actually something called the hygiene hypothesis. And so this is actually a play off of that, right? But this is kind of unique because it was looking at microbes in the soil as kind of the driving factor, right? So almost the exact same project, right? We just know a lot more and where to look and investigate now. All right, cool. So those were both really good articles, right? I really enjoyed them. One was, bacteria related. One was virus related. Again, you guys can do articles on disease. You can find other articles about cool things that microbes are doing too, but those were two good examples. So that's a good start. Sweet. My wife just found bread in the freezer, so we don't have to go to the grocery store today. Right? It's the little things. Now we're getting low on toilet paper. So, if any of you want to send a gift of non-used toilet paper, please, I'll type in my address later into the chat box. All right. The struggle is real. All right. So I have tomorrow we have, it looks like Lauren and Allie, right? Presenting their articles. So hopefully you guys have different articles. If not, you have a day to find them. And Elena, did you see the Dropbox that was up for the paper? Okay, good. Because I put it up because Caroline sent me an email because I forgot to do that, right? Just full of forgets yesterday. So the Dropbox is up for you guys to turn in your little page, summary. And please just make sure that you have the title and the author of the paper or the article that you found or used, in case I would want to look that up. All right. And if either of you for some reason didn't put that on there, just go ahead and email me that. But let's try to do that in the future for those that are coming after you. All right. So let's get started then with the rest of the metabolism lecture. We'll then move from that into fermentation, right, which is still part of metabolism. And we will also talk hopefully about carbon fixation a little bit. And we will also talk about, okay, there it is. I'm not sure why it's not showing up. Nitrogen fixation briefly too. All right, so we'll go here, there. All right, so hopefully right now you're seeing syndrome metabolic pathway, pentose phosphate pathway. You remember on your first exam, we went through glycolysis as far as we got in the central metabolic pathway. You guys had a couple TCA cycle questions, maybe one or two on there that were pulled directly off of your mastering assignments. So hopefully you noticed that, that I'm at least trying to pull a couple questions directly off of there. So, doing those and being on top of those, I think will be beneficial to you guys. Amanda, since I can see your picture right now on my little thing, because I've reduced it down to one, can you remind me of what we gain or what we're getting out of glycolysis? What are a couple things that we're getting from it? I can actually think of kind of three categories of things, but I'll take whatever you got.
So we get a net gain of 2 ATP, right?
Perfect, right? So we get a little bit of ATP. Awesome. So that's one of kind of the categories that I was thinking of. Can someone chime in with another one of the categories? What's another important thing that we're getting from glycolysis? So Amanda got the ATP for us. What else?
Pyruvic like acid. Or
Say
not
that
the
again?
pyruvates like
Okay, so yeah, pyruvate. Okay, so maybe we can do 4 categories, right? Pyruvate's the end product that's going to be important for going through a transition step before the TCA cycle. So I'll take that, right? So now there's actually 2 more left, right? So we have ATP, we have pyruvic acid. What else are we getting from glycolysis? Someone else chime in.
Whoa.
We'll let Rachel go, and then whoever else was going can do the last one.
Which Rachel? Me, Rachel?
Was Rachel's talking?
I don't know.
Rachel Stock.
Cool. The reducing power particles like NADH, FADH2, and
Okay.
NAPH, NADPH.
Well, so NADPH is actually going to come from this cycle, right? So glycolysis,
Oh, I'm on the wrong
NADH,
slide.
FADH, that's fine, right? But that's good. Reducing power, right? And that's going to be where we generate a lot of energy if we had the electron transport chain. All right, and then what's the 4th thing? There's one thing left. Someone else chime in that hasn't talked yet.
Precursor metabolites.
Precursor metabolites, right? Thank you, Lauren. And if you remember, if you go back to that chart, right, if you're a bacteria, right, you can actually use glucose 6-phosphate, fructose 6-phosphate, right? Some of that's going to go into building your cell wall. Some of that can go in making LPS, all right? Let me see my list here. Sarah, what type of bacteria do I find LPS on?
Sorry. Gram negative.
Gram negative. Yes, perfect. Nailed it, right? You guys did pretty good with that part on the exam. Oh, look, that got big. All right. So the pentose phosphate pathway is kind of interesting because glucose 6-phosphate, which is the first thing that's generated when glucose enters the cell, right? And so we may mention this later. Actually, we will mention it a little bit later down the road. But when glucose enters a cell, all right, it's phosphorylated. And what that does is that gives glucose a negative charge, right? When things have negative charges, right, it makes them hard to leave the cell. So those of you that have had AMP1, one of the things that we talked about in there is neuronal signaling, and the inside of a neuron, right, has a resting membrane potential of -70, and one of the reasons that it has that is because of sulfate and phosphate, right, negatively charged, and those negatively charged ions can't escape the cell, right, so they stay in the cell, so that keeps that cell pretty much negatively charged all the time. And so the same kind of concept here, when you pop a negative charge onto something, it makes it hard to leave the cell, and once you have glucose in, you don't want it to leave. And what's cool about glucose 6-phosphate is that it can either go to glycolysis or it can go into the pentose phosphate pathway. We mentioned this last week, but let's revisit it really quickly, right? Glycolysis is a catabolic pathway, right? It is breaking down and releasing energy. Glucose, or I'm sorry, not glucose, the pentose phosphate pathway is anabolic, right? That means it's building things, and building things cost energy. All right. So, Megan, with that basic piece of information available to you, if I gave you glucose 6-phosphate, how would you decide which way to go? Are you sending your glucose 6-phosphate through glycolysis, or are you sending it through the pentose phosphate pathway? What helps you in that decision-making process?
Knowing how much ATP is already in this.
Perfect, right? How much, right, money, right, in this case, right, ATP, right, the currency of the cell, how much do you have available? If you already have a lot of it available, you're pretty well stocked up, right? Then you're going to say, fine, go into the pentose phosphate pathway. There's things that I can build, right, that I need to invest in. If you're low on energy, well, then you're going to say, no, I can't build right now. I need to get, right, or gain energy. So you would choose glycolysis, right? So the energy state of the cell is going to be a huge determining factor in which pathway you're going down, right? You also see here, and I'll point out another one in just a second, glyceraldehyde 3-phosphate is also generated in glycolysis, and it can be shuttled, right, back and forth between the pentose phosphate pathway or glycolysis, right? And so the thing that's kind of cool about both of these pathways is that there's not necessarily a strong commit, all right? So you could start building stuff and then realize you don't have enough energy, right? And shuttle it back to glycolysis where it can be broken down so energy, right, is released. Opposite is true. You could be breaking things down to gain energy because you think you're low, and it actually turns out you have enough, so then you can shuttle back to the pentose phosphate pathway, right? So there's a lot of kind of options or interplay between these two pathways. If you go back to that original diagram a few slides ago, I think it's a kind of important reason that it shows the pentose phosphate pathway kind of off on the side, all right? Because things can be shuttled back and forth. It's not a, you know, you're locked into this sequential order when you talk about these two pathways. So this is what Rachel Stock actually mentioned, right? NADPH. So NADPH is generated here in the pentose phosphate pathway. This is the only place that it's generated, right? And the reducing power that we actually get from it is invested directly back into anabolic reactions, all right? We'll talk about that here in just a second. So the pentose phosphate pathway, and you'll see this again here in just a moment, generates two important metabolic precursors, ribose 5-phosphate and urthorose 4-phosphate. And these are going to be used to build nucleic acids and amino acids, right? So when you think about the pentose phosphate pathway, you really need to think about anabolic, all right? It's not generating ATP, or does not lead to even the indirect generation of ATP that you can invest in things later, right? It is interested in making precursor metabolites, right? And then using the reducing power that's generated there to start building things. So we've already kind of mentioned this, right? It's anabolic as opposed to being catabolic, all right? And some of the things that we can generate and build: fatty acids, nucleic acids, amino acid synthesis, right? So if you're thinking about the macromolecules that are essential to life, right? I can even add carbohydrates to here because you can see that they can generate 5 carbon sugars, right? So the macromolecules, right, that you need, that bacteria need, are going to be generated in the pentose phosphate pathway. It's an important way that we can generate them and build them, right? So here, right, we already mentioned glyceraldehyde 3-phosphate, which is this. Here's also fructose 6-phosphate, right? And you could even you could say glucose 6-phosphate here, right? So these are a little bit farther down than glucose 6-phosphate. This is almost directly after it. This is a little farther down, right? So again, you have this ability to, once you've started, if the demands of the cell change, you can shuttle back and forth, right? So really important feature between glycolysis and the pentose phosphate pathway. Within the pentose phosphate pathway, you kind of have two major phases, or you could even argue two major branches, right? So you have the oxidative branch, which is generating NADPH, right? And then you have the non-oxidative, right? So this is going to be the generation of 5 carbon sugars. And these 5 carbon sugars can then be used to build various macromolecules that the cells need, right? Again, this can be you. This could be bacteria. Okay, I already said that. This just kind of shows you this general process. And so for every molecule, right, of glucose 6-phosphate that is entering the pentose phosphate pathway, right, you're generating 2 molecules of NADPH, right? So you can see both of those right here. rest easy. I'm not going to expect you to draw this out. I'm not even directly interested in either these enzymes that you see here, 6-phosphogluconolactonase, right, and 6-phosphogluconate dehydrogenase. But I at least want you to appreciate, right, we talked about this, or we started to mention this, that every step in a metabolic pathway, right, is having some type of enzymatic regulation, right? So you need the presence of these two enzymes for these things to happen. Things aren't just going to fall off. Things aren't just going to be added, right? So this is enzymatically driven, all right? So this NADPH is generating reducing power that can be used, right, to build molecules, all right? And, you know, this is just up here to show that NADP, I don't know if you thought about this, but it kind of makes sense, is considered a regulatory step, right? Because if you don't have NADP, well, then you can't carry out this reaction, all right? And this goes back to my bippity boppity, science is not magic, right? Electrons, protons, they have to have places to go. And so if you don't have acceptors, well, then the reaction can't function. And that's an important concept to kind of plant in the back of your mind, because if you are capable of the electron transport chain and oxygen is your terminal electron acceptor, and you don't have oxygen, you don't have that last acceptor, so then you can't continue or finish the process, all right? So we'll kind of visit that, right? Irreversible, that's fine. Again, everybody, but Megan's the picture that I can see right now, because she answered the last question. Everybody but Megan has to memorize, or doesn't have to memorize this, right? So that means Megan has to memorize this. Right, Megan, you cool with that? Good, she's up for the challenge. No, I'm just kidding. So this is, again, showing you that non-oxidative pathway. I don't expect you to have all of this memorized. There's a couple things that I want to point out here, though, right, is that here you see glyceraldehyde 3-phosphate, here you see fructose 6-phosphate. You can see 5 carbon sugars, right, being formed. You can see a 4 carbon molecule being formed down here. Again, these are going to be used to start building the macromolecules that bacteria need, the macromolecules that you need. But you see here, right, that glyceraldehyde 3-phosphate, fructose 6-phosphate, right, I highlighted those because, again, they can be, right, shuttled back into glycolysis if, right, you're needing to further, right, break things down so that you can release the energy that's associated with catabolic reactions. And so again, I know this looks kind of busy. I'm not expecting you to take a lot away from it, other than here you see, right, some 5 carbon sugars. Here you see some 4 carbon molecules. So those can be used to build, but then you have this glyceraldehyde 3-phosphate, fructose 6-phosphate that can also, right, be shuttled back to glycolysis if we need more energy, right? So I love these pathways. I think commitment is very important in life in various aspects, but it's also nice to have some flexibility, and you see flexibility within these pathways, all right? So if you're kind of freaking out, which I don't think you are, but you could be, and that's fine if you are, at the end of these, you know, central metabolic pathways, right, I have these kind of summary slides. And so what do you ultimately need to know? Now, if you know everything on this slide, I'm not saying that means 100% on the question, but that means that you probably will get a majority of the points on the question because you understand the basic concepts, all right? So pentose phosphate pathway, right? The only source, all right, you got this, Rachel Stock? The only source of NADPH, right? And we call this reducing power for a biosynthetic, right? Synthetic, you're building. So this is reducing power that is used, right, directly in anabolic reactions, all right? So particularly nucleotide and amino acid, but you can also build other macromolecules from precursor metabolites that are generated in this pathway, right? Regulated, right, at the committal of glucose 6-phosphate. And so this is kind of, hey, you have to decide early on which pathway am I sending this glucose 6-phosphate down? And remember, glucose 6-phosphate, that first, again, I'm not going to say regurgitate this enzyme. If you want to, that's fine, right? That's the first step in generation of this reducing power. But remember, the thing that's nice, right, is that you can feed back, and I probably should have added this, so make sure you have this in your notes, right? With fructose 6-phosphate and glyceraldehyde 3-phosphate, right, can be shuttled back. The primary source of 5 carbon sugars from nucleotide synthesis, right? You can see that up here. And here's that glyceraldehyde 3-phosphate, here's that fructose 6-phosphate, right? So this ability to work backwards, right? And if you start working backwards, you go back into glycolysis, and then you can continue on to the TCA cycle, right? If energy acquisition is the name of the game that you're in. All right? Cool. Gloria, how do you feel about that? Okay, good. Those of you that have had AMP1, you know, I think this is a pretty kind of good build off of some of that information. I can't remember. Toomey, can you remember, do we do this in Bio 1? Do we do some cellular respiration stuff?
When I took Bio 1, they didn't teach us respiration, but a semester after I finished, I was trained for Bio 1 and they started teaching respiration.
Okay, there was a change, and so yeah, I haven't done Bio 1 in a couple of years, and so I couldn't remember. So maybe you got it there. If you took it at the same time or before Toomey, then you didn't get it there. So, you know, be sure to look over this. And again, I know some of this you guys already did, you know, some questions related to this, because I thought I'd be a little farther along. So this is a section, though, of your textbook that's worth reading through. If you still have some questions, and please reach out to me. It's often my opportunity to remind you. And, you know, based on your test scores, there wasn't anything major, but please, if you have questions on the material, right, shoot me an email. I can answer questions that way, or we can arrange a Zoom meeting. All right. Now, after glycolysis, right, we can then enter the transition step, which connects glycolysis and the pentose phosphate pathway, again, because the pentose phosphate pathway can shuttle back into glycolysis, connects them to the TCA cycle. And so, right, one of the major events that's occurring here is we're taking pyruvate and converting it into acetyl-CoA. Again, please pay attention here. Why do I have pyruvate generating 2-acetyl-CoA? Why does that happen? Why do I have 2 there? Megan?
There are 2 pyruvic acids that go in.
Perfect,
because you split
right?
the glucose.
Yes, I love that, right? And again, bippity boppity, biology's not magic. And so you started with a 6, right, carbon glucose. You've chopped and diced and sliced and miced, right? And you now have 2 3-carbon pyruvates at the end. So each pyruvate's turned into acetyl-CoA, right? So you have pyruvate at the end, 2 of them, so that means you get 2-acetyl-CoA, all right? And notice here, even this transition step, we're generating a little bit of reducing power that's eventually going to help us out with generation of energy further down in the electron transport chain. Okay. Don't expect you to memorize this necessarily, right? But remember, we took pyruvate, we made acetyl-CoA. So this is the CoA component of acetyl-CoA. And the reason that I put this on here to highlight, I know we don't spend a lot of time memorizing different enzymes and doing all this stuff, but what I want you guys to appreciate is that if this CoA was not attached to the acetyl group, enzymatic recognition in the TCA cycle would not be possible. So basically, you are tagging that acetyl group, right, with this CoA molecule so that enzymes in the TCA cycle can recognize this structure. You knock off CoA, enzymes can't interact with acetyl, you then don't generate a bunch of reducing power that you'll see here in just a second. All right. So this is kind of to Megan's point a little bit, all right? For every glucose, right, there are 2 cycles of the TCA cycle, because for every time you go through glycolysis, you are generating 2 molecules of pyruvate, right? And one of the big things here, and I'd actually say go ahead and add if you're keeping notes at home, but you'll see these NADH, which are also generating a lot of FADH, all right? So the TCA cycle is incredibly important in the production of reducing power that will be used later during the electron transport chain, if you have one. All right. Let's see, I have my highlights, yes. So this is the TCA cycle. So a couple things that you can see here. First of all right, you can see alpha ketoglutarate, oxaloacetate were actually 2 precursor metabolites, right? So you could go back and look at that diagram. That's one of the first couple slides, or that little table. That's one of the first couple slides in this unit. You see these are precursor metabolites. You also see here NADH, NADH, all right, NADH, FADH2. So you can see a lot of reducing power being generated. As before, all right, you are generating some ATP, but not much, right? So for every molecule of glucose, you get 2 ATP, a net gain of 2 ATP from glycolysis. For every molecule of glucose, you also get 2 molecules of ATP from the TCA cycle. So if you look at the central metabolic pathways, because remember, pentose phosphate's not generating ATP for you to use. So that means that you're left with glycolysis and the citric acid cycle. You're only getting 4 ATP. Does anyone remember, again, kind of estimate off the top of your head from a previous biology class maybe, or previous A&P 1 class, how much ATP is generated in cellular respiration?
So like 34?
Yeah, 30-ish, right? Because you're adding some of this on, so that would be kind of 34. So you're generating somewhere around 30 molecules of ATP via cellular respiration, right? And if you're saying 34, and again, the estimates kind of vary. Sometimes you'll see 32, 34, maybe even up to 36. It's not actually an exact science. But, you know, what we can tell you is that very little of that ATP that you're getting at the end of the road is coming from these 2 central metabolic pathways, all right? So most of it's going to be generated later because of the reducing power of NADH and FADH2. The reason I highlighted this, and you guys will see this test kind of soonish, is that some bacteria actually use citrate as their carbon source. That's kind of a unique feature or function. Most bacteria prefer, right, 6 carbon sugars, specifically glucose is kind of the again, one of the most sought after nutrients. And in this case, right, certain bacteria have evolved and developed an enzyme called citrase. And citrase breaks down citrate into carbon molecules that can be utilized for cellular work within different bacteria, right? And it's a really simple test, but it's a really powerful and a test that's very reliable. So we'll talk about that a little bit more. That's why that's highlighted. I try to point out some correlations between these different things and what you see in lab. Unfortunately, you won't be seeing quite as many of them, but I'll try to give you guys a heads up on some of those. All right. So TCA cycle, right? Again, the summary slide. 2 ATP, not much, but 6 NADH, 2 FADH2. Again, this is for every molecule of glucose and some of those precursor metabolites. So the TCA cycle, right? Reducing power, reducing power, reducing power. It is going to generate a lot of energy further down the road. All right. So this is a table from the textbook that I previously used, but you should find something similar in the online textbook that I hope you're at least, you know, utilizing and looking through at a minimum for the assignments that you have due, you know, kind of the day or the morning of class. But you would see something like this, right? So it gives you these different steps and kind of, hey, what's kind of being generated in there, right? Some of the things I want you guys to have a little bit more detail than they have here, but this is a good starting point, right? Please remember, I love these tables. If you just memorize them, you're going to get some points, you're not going to get them all, but if you're using them to help you study, these are always good jumping off points. Right? So the electron transport chain, all right? I kind of like to think of tracks, right? This is a train that could be in trouble. Presumably what's going on here, right, is they're actually just having the train be stored here. But, you know, yikes, be careful, right? Finish the bridge. So let's talk about the electron transport chain. So when you hear about cellular respiration of the electron transport chain, one of the things that you may see or hear or read is this term oxidative phosphorylation. And oxidative phosphorylation is actually referring to two things. It's referring to the actual electron transport chain and the molecules and proteins that are associated with it, which we'll talk about. And then, right, it's also referring to the actual generation of ATP. All right. We'll go with Olivia. Do you remember, all right, where the electron transport chain, if you have one, is in our cells and where it is in prokaryotic cells? Is it the same? Is it different? What's going on there?
Is it in the mitochondria of our cells?
It's in the mitochondria of our cells, perfect. What about in prokaryotic cells? Where does it occur? I'm totally fine if you want to phone a friend.
Yeah, I'm gonna need to phone a friend.
Phone a friend. Let's see. You know, one of the things that I miss about lecture is that in lecture, I could smell fear, right? And so that's who I'd call on. It's a lot harder to smell over the internet. Rachel Dickey, can you help me out? So it occurs in mitochondria in you. Where does it occur in prokaryotic cells?
The membrane.
The membrane, right? It's part of the cytoplasmic membrane. So if we're talking about the mitochondria, right, you could say it's the battery that powers the cell, right? I know that's not, you know, maybe satisfactory to people that really love the mitochondria, but that's fine. It's a good, simple comparison. Whereas the whole bacteria, all right, is a battery, all right? So good, cytoplasmic membrane of a prokaryotic cell, mitochondria of a eukaryotic cell. This is kind of simple. Maybe you've seen this diagram before. I do kind of like the simplicity of it. So here you see an electron. This is the start of the electron transport chain. This is the end of it. Kind of think of it as steps. As it moves down, we end up generating more ATP. And then at the end, you have to have a terminal electron acceptor, which in this case is going to be oxygen. All right. One of the things, oh yeah, see, electron transport chain, it's fun. One of the things that I don't like about the simplicity of this diagram is it doesn't, I think, do a great job of reminding you of the fact of, you know, if you look at this slide, right? So when I ask questions too, please don't be afraid to read the slide sometimes. Now, just because I ask a question doesn't mean the answer is on the slide, but that's a good place, you know, to start. So Allie, what, let's go back. Okay, fine, be that way. Allie, what is traveling with electrons? What always goes with them, right? They don't like to go alone, so it's always with them.
I'm not sure.
Allie, what's
Oh,
always
protons.
protons? Perfect, right? So a proton, right, is a fancy word for a hydrogen ion, right, which has a positive charge. And I want you to plant that idea in your head, right? This is really important, right? So as the electrons move through the electron transport chain, protons are moving with them. All right, so just kind of file that away. All right, we'll get back to why that's an important concept here in just a second. So for the electron transport chain, you basically have two categories of carriers, right? You have large complexes, all right, that some, not all, and you'll see this in a second, have the ability to serve as proton pumps, all right? So again, planting that idea, well, where are the protons coming from? How can the proton pumps do their job? Well, the proton pumps are doing their job because the electrons are carrying protons with them. So you'll see that in just a second. Now you also have smaller compounds, and you can actually see those right down here, which we'll talk about, that are moving electrons between these major complexes, right? So you think about it how you will. I think about these small compounds that are moving electrons kind of like taxis that are getting the electrons and the protons from point A to point B. All right. So three types of electron carriers, quinones, cytochromes, and flavoproteins are going to be transporting these electrons with protons between the major complexes of the electron transport chain. And you'll see what those complexes are here in just a minute. So for the quinones, right, you have different types of quinones. Two of the major types are ubiquinone, which you have present in your cells, and then menaquinone, which is going to be actually present in bacterial cells. Now, bacteria, some can have ubiquinone, all right, and some can have menaquinone, right? So it's not like these are exclusive to prokaryotic cells and these are exclusive to prokaryotic cells. I'm sorry, it's not like these are exclusive to prokaryotic cells. These are exclusive, right? You don't have menaquinone as one of your quinones. So this is a unique quinone that we find in prokaryotic cells, right? And it's this quinone, right, that is actually used to generate and make vitamin K. And so your gut bacteria are actually an important source of vitamin K, and vitamin K is needed for coagulation of the blood. Laura, what's coagulation?
Like when it all sticks together and gets kind of clumpy.
Right, it sticks together, right? So you've heard of a blood clot, right? Coagulation happens for that. So this is, right, when you get cut or you have an injury, you don't bleed out. And so the bacteria in your gut, right, are actually an important part of, or important source of vitamin K, which is needed in the coagulation or blood clotting process, all right? So I just plant reasons throughout this whole course for you to love your gut microbiome, all right? It's the best. And remember from that presentation, there are 10 times more bacteria in your gut, right, than cells in your body. So for a cell-to-cell basis, you are more bacteria than you are you, right? Now, quinones are lipid soluble, allowing them to move through the membrane. Remember, if we're talking about bacteria, right, electron transport is happening within the cytoplasmic membrane. If we're talking about our cells, it's happening in the mitochondria, which is a double membrane bound organelle, right? So you need something that can move through fats because membranes are made of fats. All right, so as I mentioned, this transfers the electrons between some different complexes, so we'll get to in just a second. All right. Other electron carriers, right, are the cytochromes here. Cytochromes contain a heme group, which is iron. All right. So again, this is a reason that iron is important in the electron transport chain. For the most part, you guys did a pretty good job of at least kind of giving me an idea of why iron is important. All right, and so here you can see it's important in the electron transport chain. And so if you're a pathogen, right, that relies on the electron transport chain to generate energy, this gives you another reason why you're trying to steal iron from the host. All right, there are multiple cytochromes that exist within different organisms. They're usually designated by a letter, right? Usually alphabetical, A, B, C, D. And different species have different cytochromes present. Some bacteria have an enzyme, cytochrome C oxidase, that's part of the electron transport chain. You guys actually do, or would do if you were in lab, an oxidase test, and that's looking for the presence of this enzyme. And so if this enzyme is present, then you can feel confident that that bacteria has the electron transport chain. So Neisseria, Pseudomonas, Campylobacter, these are all cytochrome C oxidase positive. Now, just because an oxidase test is negative doesn't necessarily mean that bacteria doesn't have an electron transport chain. It just means that cytochrome C oxidase is not part of the electron transport chain. It would have a different, or could have a different cytochrome present. All right, but again, a taxi cab kind of carrying the electrons, just like ubiquinones or quinones. All right, the last carrier, right, that's working between these complexes is flavoproteins, right? And flavin, right, you've seen this before, so the F in FAD is actually a flavin. And those of you that are health nuts maybe take vitamins, right? So vitamin B2 is actually riboflavin. And so you're actually putting flavins into your body, right? And these can be used in generating FADH, right? Or they can be used in generating flavoproteins that help transport electrons, all right? So in this case, right, we're not seeing anything here that's necessarily unique to prokaryotic cells. All right, so let's kind of we'll do this, and then I'll give you guys a little bit of a break, right? So I just want to do kind of a comparison between the electron transport chain in eukaryotic cells and prokaryotic cells. So those of you that have had some type of biology or AMP in the past, this may be familiar to you. So a couple of things I want to point out here, all right, is that what you're seeing, right, is this is the phospholipid bilayer within our mitochondria, right? So this is taking place in your mitochondria. So this is actually up here. This is the inside of the mitochondria, this intramembrane space, all right? So this is actually the inside of the mitochondria. So here you have NADH, right, that you generated in glycolysis in the TCA cycle. It's dropping off its electrons. And when it drops off electrons, right, it's carrying protons with it. And your complex 1 is actually a proton pump, so that's why you see protons coming out here. Complex 2, right, is not a proton pump, all right? It is simply kind of a go-between, all right? Here you can see ubiquinone, cytochrome C, right, present in this case. What they don't show here is that FADH2, right, is actually dropping off its electrons at complex 2. If you want to get really, if you want to start to go to more in depth in this, you can actually do a calculation of energy that's generated by NADH and FADH2, and you actually are able to generate slightly more energy with NADH because it's dropping off its electrons and protons farther upstream in the electron transport chain. All right. You then go to complex 3, which again is a proton pump. Here's cytochrome C transporting in between. Here's complex 4, which is also a proton pump. You also see oxygen here as a terminal electron acceptor. And all of this is used to generate a proton motive force. So basically you're getting a big concentration of protons on this side of the mitochondria, all right? So you now are kind of gonna basically rely on diffusion, high concentration out here, low concentration here. So these protons rush through this complex called ATP synthase. Depending on how you've been taught or where you learn, some people even call this complex 5. I'm fine with you calling it with whatever you want. As protons move through this complex, they basically cause it to kind of spin like a windmill. And when it spins like a windmill, it's basically taking phosphates and popping them onto ADP, right? Which is missing one phosphate. So then you generate ATP. So what it's telling you here is that for every 10 protons, you generate 3 molecules of ATP, right? So that's why I really was trying to emphasize, hey, when electrons are moving, that's super important, right? And the reason it's important is because they're carrying protons with them, and we're using to generate this force right here, all right? So this is the one thing you're doing with that proton motive force. Here is everything that I basically told you, right? 3 of those complexes are acting as proton pumps. Remember, complex 2 is not. You have 2 major electron carriers, ubiquinone and cytochrome C. And here, right, is everything that I essentially just told you. Here you see complex 2, FADH, is dropping off there. So yeah, so go ahead and maybe kind of look through this, but this is everything that I basically just told you, all right? Now, here is the electron transport chain in prokaryotic cells. Hopefully, you can notice right away, right, that there are some kind of important differences that are present. So let's kind of walk through this really briefly. One of the things I want you to notice is that they are actually missing one of the major complexes. They don't have it, right? So we actually have an extra proton pump that's not present in prokaryotic cells. Same basic principle though, their NADH dehydrogenase is the equivalent of our complex 1. Right here, succinate dehydrogenase, which is the equivalent of our complex 2. Just like our complex 2, right, this is where they are receiving FADH2. Here they have ubiquinol oxidase. We'll talk about that a little bit more in just a second. Oxygen is a terminal electron acceptor here, right? That means this whole thing can run. And so here, right, you see you're pumping out these protons. Now, please note that the proton motive force, that proton gradient, is now being used for multiple things. So again, ATP synthase, just like we would do, we actually started to talk about, right, flagella here, right? So remember, flagella are being powered by the proton gradient. And here we also see the proton gradient being used to actively transport molecules, right? So in this case, right, you are going against the concentration gradient with whatever this transport molecule is, and it's basically tagging along with these protons, right, that are rushing in because there's a lot of protons out here, there's not many down here, all right? So they are using the proton gradient to help actively transport, right? This is an energy saver. This is something that we don't do. So our proton gradient is used for one thing. Prokaryotic cells are actually using it for 3 different things. All right. Oh, and note, you know, it doesn't really say it here, but this is the actual cytoplasm. So this is the inside, right, of the bacteria. And this is the outside right here. All right. Now, again, this is not just it's floating off into space, right? This would either be that periplasmic space or that gel-like layer, right, if you're gram positive. So kind of keeping it tight to the vest. So again, 2 kind of major differences. Now, let's kind of briefly go into some other important things. So aerobic respiration, right, is saying the electron transport chain is taking place in the presence of oxygen, right? That's exactly what we do. So kind of same thing there. You saw some of the components are slightly different, but the overall concept is the same. What becomes really kind of cool or unique is that some bacteria are capable of anaerobic respiration. That means oxygen is no longer the terminal electron acceptor. That means they're capable of using the electron transport chain, right, or having an active electron transport chain in the absence of oxygen, right? And we'll kind of continue to talk about some of the consequences that occur because of that. But that's really cool, right? If I take oxygen away from, I'll take it from Amanda, right? That's bad, right? And it's bad for multiple reasons, but one of the reasons it's really bad is that then takes away the ability of her cells to generate a sufficient amount of energy that she would need to live, right? We'll talk about, and you guys can do this too, there is the ability to generate energy in the absence of oxygen, but it's not nearly as efficient, and for us would not be sustainable in the long run. All right. So, you know, when we're comparing, you know, these different transport chains between the two, right? So prokaryotic cells have two different NAD dehydrogenases. This is their complex 1 and their complex 3. So their first NAD dehydrogenase is equivalent to our complex 1. Succinate dehydrogenase, right, which I told you, which is not a proton pump, is equivalent to our complex 2. It's merely an electron acceptor. They are lacking, right, our complex 3 equivalent. So their third, right, kind of major acceptor, their second proton pump, if you want to look at it that way, is the equivalent to our complex 4. Now, what gets really interesting is that their last proton pump, right, their complex 3, our complex 4 equivalency, there's some unique variation here because they, right, have the ability to drop off electrons sometimes to other terminal electron acceptors besides oxygen. Our complex 4, right, our last proton pump is designed to interact with oxygen. Bacteria that have different terminal electron acceptors, right, have a variation in their final proton pump, allowing them to drop off their electrons there. Now, there is a consequence, I guess if you want to say that, for anaerobic respiration, and that is the terminal electron acceptor, right, if it's not oxygen, is not nearly as hungry, we'll say, for electrons. That means it's not quite as excited to take them. The more excited something is to accept the electrons, like oxygen, the more energy we can generate during that process, all right? So terminal electron acceptors that are not oxygen have lower electron affinities. They're not as willing to take those electrons, so you're not going to generate as much energy. So if you go down to the second bullet point, here's what I mentioned, right? If you are dropping off your electrons at a different terminal electron acceptor, well, then you're going to have a unique variation in your last proton pump that we don't see in our cells. Some other terminal electron acceptors can be nitrate and sulfate. These are used by different bacteria, typically bacteria that you find in kind of unique environmental situations. All right. And I think at the end of this presentation, or somewhere, I have a chart that kind of shows your electron carrier to your electron acceptor, the electron affinity for them. The larger that gap, the more energy you generate. So let's say this is like NADH, then oxygen is all the way down here, all right? If you use a different terminal electron acceptor, the gap between the electron affinity between the two is closer, and that means you're not going to generate as much energy, right? So we already went over these in our last, or a couple slides ago, so please just remember what are the different things the proton gradient is doing in prokaryotic cells compared to eukaryotic cells. This is just showing you basically the idea of protons will rush in, right? They'll cause complex 4 to turn, and during this process, right, they're popping phosphates onto ADP to give you ATP. This is just showing you, right, the energy yield between NADH and FADH2. So you see for every molecule of NADH, you're getting about 2.5 ATP, right? So 10 times 2.5, 25. For every molecule of NADH, right, you're getting about 1.5. And again, this is not because NADH is a better electron carrier. It's because, right, it's dropping off at complex 1, which pumps protons out. FADH is dropping off at complex 2, which is not pumping protons out. Remember, protons, every 10 of them give you 3 molecules of ATP. So the more protons that you end up having pumped out means you're generating more energy, right? And so again, I say 32 here with the total. You can also see 34 up to 36 depending on the variation. But again, the point being, this ATP count, this is coming from those central metabolic pathways. So most of the ATP that you get is from the reducing power that's generated in those pathways. All right, this is just rule of ATP if you need to look over basics. This is what I was referring to for electron kind of affinity. So here you have your organic compounds, in this case, glucose, that were more than likely breaking down. And so you're going to put those electrons and protons on NADH, right? Here's oxygen all the way down here. So you can see there's a big gap. That big gap is allowing the generation of more energy. If you go to sulfate up here, or here's nitrate and nitrite, which are also acceptors, you can see the gap's not as big, so the energy generation is not as efficient, but it's still, right, an important function that's allowing you to generate a good amount of energy in the absence of oxygen. Basic enzymatic principles, if you need to look over that. This is just some chemistry basics. Some of you guys are good on that, but it's just there for you guys to review some of that stuff, right? So what we're gonna do is, it is 9:15ish. Let's come back at 9:30. We'll go through fermentation and carbon fixation and nitrogen fixation, more than likely, relatively quickly. All right, so I'm just going to go ahead and leave this on. You can stay on if you want. You can get off if you want. Just make sure you're back on at 9:30. Okay. Nitrogen. Go ahead and get started back here. These next little bits are kind of small, so they shouldn't take too much time. This is a really important part of the metabolism chapter, though, because it focuses on fermentation. So what's going to happen when organisms are incapable of cellular respiration, or something is inhibiting their ability to generate energy via respiration, right? And that's where fermentation comes into play for us. So, a couple different things, and this, I could even say that we can even slightly modify this, right? We need oxygen, right, to generate enough energy to sustain ourselves. Prokaryotic cells, that's not necessarily always the case, right? And we even talked about some of the different oxygen requirements that they have in the previous unit, right? It's also important to note that not all prokaryotic cells even have an electron transport chain. So every cell in your body, right, has an electron transport chain. That's not the case for prokaryotic cells. And so that's just the way that they function, all right? And so when it comes to fermentation, what I really want you to kind of hone in on is that organisms ferment for basically 2 reasons. They don't have a terminal electron acceptor, which in a lot of cases is oxygen, but is not necessarily always oxygen, right? So no terminal electron acceptor, right? It's not present for whatever reason. And or, right, they don't have an electron transport chain. So it doesn't matter if something that you would say is a terminal electron acceptor is present. If you don't have an electron transport chain, well, then you're going to go through fermentation anyway. All right. So one way to look at fermentation, right, if you want the fancy definition right here, it says metabolic process that stops short of oxidizing glucose or other organic compounds completely. That's a fancy way of saying that you don't kind of finish the complete breakdown of that original glucose molecule like you would if you were going all the way through the central metabolic pathways to the electron transport chain, right? So you're not kind of finishing the entire process that we associate with cellular respiration. For organisms that are undergoing fermentation, right, ATP is coming from glycolysis. If that's the case, Anna, how much ATP am I generating from each molecule of glucose if I'm just relying on glycolysis?
I want to say 2.
I want to say you're right. Perfect, right?
Okay.
And so you're generating 2 ATP if you're generating ATP solely through glycolysis. So you could certainly see, especially larger organisms such as ourselves, how this is not a sustainable process. Now, those of you that have had AMP1, we talked about this idea of being able to do this temporarily, right? If we refer specifically to muscle contractions. And so when blood flow is cut off to muscles during a strenuous workout, or when oxygen levels are running low because of a strenuous workout, your muscles are able to generate ATP via glycolysis, and that leads to the formation of something called lactic acid if we're talking about your muscles. And this is a good temporary fix to get you through the workout or get you through whatever activity you're doing, right? But it can't be a substitute for prolonged function, right? Or can't be used for prolonged function. Even though we're only going through glycolysis, we still have the reducing power, NADH, right? So we're still generating, right, protons and electrons. They still have to have somewhere to go. And so NADH is still really, really important there, all right? And in this case, right, NADH is picking up an electron and protons. It still has to have somewhere to go. And so now, right, pyruvate, or a derivative of pyruvate, is your new terminal electron acceptor. Right? We're not going through that whole process. We're not going through glycolysis. We're not going through the TCA cycle. We're not going to the electron transport chain where oxygen, or in the case of bacteria, something like nitrate, or sulfate are the terminal electron acceptors. Pyruvate, right, that 3-carbon molecule that you generate at the end of glycolysis becomes your new terminal electron acceptor, right? And when pyruvate or its derivatives become your new terminal electron acceptor, we have some interesting things happening, right? And what we see, right, are those sugars or starches or carbohydrates that you normally would have taken all the way through the process, and NADH, FADH2, right, drop off that's not happening anymore. And so now when pyruvate accepts, right, we start to generate alcohols and other organic acids. All right, and you'll see kind of an example of that in just a second. Now, what's really cool about this is important in food processing. This is also important in your body. And so when fermentation occurs, right, 2 things are happening. One, you're removing energy sources from harmful microbes. And so although you don't take these things all the way through cellular respiration, if you start to chomp or eat them up, that takes them maybe away from bacteria, potentially pathogenic, that would. You also, by generating alcohols and acids, are creating an environment that is inhospitable. All right. So, a great example in the human body is actually the female vaginal tract, right? And it actually contains a high level of lactobacillus. And the lactobacillus in the vaginal tract of females, right, produces lactic acid, which lowers the pH of the vaginal tract, which actually helps keep out bad bacteria. All right. It's also the reason that semen from males contains an alkaline solution to try to balance out that acid, because it's great for keeping bad bacteria away. It also, though, kills sperm. All right. And so we see in semen the introduction of alkaline solution to combat the acidic environment of the vagina that exists because of bacteria, right? So it is a very beneficial thing, even for us as humans, right? But again, this inhospitable environment could be for some of your favorite fermented foods, right? It keeps away the bacteria that would spoil or make those foods go bad, or even keep away the fungi that would make those foods go bad. All right, so if you like yogurt, we're at Lee, so I don't know what this picture is. It just happened to show up when I looked for fermentation in food and beverage products. All right, maybe you like pickles. Maybe if you're of Nordic descent and you like pickled fish. I don't know. From the South, some of us like our hot sauce. If you like cured meats, right? These are all relying on fermentation. And fermentation for us is nice because it gives it kind of a unique, fun taste for a lot of these different things, but it also is a great preservative. Truth be told about this beverage called beer, which I'm sure some of you are seeing for the very first time, when explorers and settlers were traveling across the ocean, right? Fermentation and the production of beer was actually really important because the microbes and their byproducts would actually help remove some of the bad stuff from the water. And so this actually, right, resulted in a beverage source that was not going to harm them like contaminated water could. All right. And there's other things that you could think of, right, that are fermented. I heard there's this other beverage called wine. Apparently my bro JC made it from water at a wedding party once, right? So again, we know of fermentation kind of all throughout our lives, all right? So again, organisms can't go through cellular respiration. Man, I'm mentioning this over and over again, so this could be a good test question, right? Fermentation is occurring because of a lack of an inorganic terminal electron acceptor or oxygen, or I'm sorry, an inorganic terminal electron acceptor, which we often say is oxygen, but could be sulfate, right? Could be nitrate, nitrite, right? You have a couple different things there. So no terminal electron acceptor, or, right, a lack of an electron transport chain. Now, some microbes, right, have the ability to switch between respiration and fermentation, right? So they much prefer cellular respiration as a means of generating energy. But if for some reason that terminal electron acceptor is removed for a time, well, they can generate enough energy via fermentation, right? And if we were in lab, you would see this with some of the different bacteria that you guys would work with. Fermentation of glucose. All right. So you see here, energy generation occurs during substrate phosphorylation during glycolysis, right? So this is kind of that substrate phosphorylation. Again, is another way of saying incomplete oxidation. You're not breaking down glucose to the extent that you would if you were going through all those central metabolic pathways and you had an electron transport chain waiting for you at the end. And so this chart, again, maybe you love it, maybe you hate it, maybe it means nothing to you, that's totally fine. I like it because it just shows that if you're breaking down something like glucose and pyruvate is your terminal electron acceptor, well, that means it's another organic compound. You can see here, these two boxes are almost side by side. And side by side, right, from glycolysis, Anna told me she's generating 2 ATP. They're so close together, there's not a lot of ATP generation compared to you breaking down glucose and then having something like oxygen as a terminal electron acceptor. The gap between them is great. That means that the energy that you're going to generate is great. But this is only occurring, right, if you have an electron transport chain. Now, again, let's bring NADH and pyruvate back into the fold, right? So I told you pyruvate or derivative is acting as a terminal electron acceptor. It's taking the place of oxygen or whatever your terminal electron acceptor might be if you're one of those bacteria that has an alternative. So, right, what's going to happen when you start to break down glucose is you generate NADH. NADH, right, plus an extra proton it's carrying. So that means you started with NAD, you break down glucose, it picks up an electron and some protons. Just like if you had an electron transport chain, NADH, right, plus this extra proton that it carries, it wants to drop it off, right? It wants to deliver it to something that wants it even more than it does. And in this case, right, it drops it off at pyruvate. And when you drop that off at pyruvate, right, you now see the generation of lactic acid. All right. So in this case, right, you see the two protons are being dropped off on the second carbon. So here's the switch, right? So pyruvate and lactic acid, right, are almost identical molecules, except you see this second carbon has the hydrogens that came from NADH. In some cases, pyruvate can lose a molecule of CO2, right? So that pyruvate derivative still accepts electrons from NADH, right? So that kind of intermediate is acetaldehyde. When acetaldehyde accepts electrons, it then leads to the formation of ethanol, all right? There are other acids that can be generated. You'll see some of those. They're all kind of variations of this process. But again, pyruvate or a derivative of pyruvate accepts, right, electrons and protons from NADH, and then that leads to the formation of these biochemical byproducts that we then utilize for our food sources, or our bodies may utilize to help regulate the bacteria that live in certain biomes. Okay. So these end products, right, if we are in a laboratory setting, a clinical setting, right, a research setting, are important for helping us identify different isolates of bacteria. Some bacteria, first of all, can ferment, some can't. So if you see the presence of acid, you know they're capable of fermentation. Some of the acids they produce actually vary. And so depending on what's produced, acid, ethanol, certain gases even, that can help you identify them. These products can be commercially valuable. We'll actually talk about that just briefly here and not too long. And, you know, again, here's some of the different common end products. I'm not going to spend time on this because you'll see on my next diagram, those same end products are listed. And so here, if you have pyruvate, right, what's this showing is pyruvate. It's saying pyruvate is kind of your parent molecule, depending on the derivatives that you have coming off from it. These are the different end products that you're generating. So lactic acid, ethanol, butyric acid, propionic acid, mixed acids, butanol. You can see some of the other end products that exist. Not only do you have ethanol, but you also have CO2 sometimes as a byproduct. Here you see CO2 is pretty common. Even hydrogen gas can be common. And so you're using some of these different things for a benefit. Here you see a lot of food sources. Here you see them using as chemical tests. Here's acetone. Those of you that paint your nails, right? Acetone is the main component in nail polish remover. So you've probably used that in some form or fashion. Isopropanol, right? And ethanol can be, right, common disinfectants, which again, if you think about beer and wine, right, the alcohol in them is ethanol, and that is helping control some of the stuff that wants to spoil them. So when I say not just for food, some bacteria, this is actually from some Clostridium, are capable of something called ABE fermentation, right? That means acetone, butanol, and ethanol. They can produce all of these things. And what's really cool is this is a guy named Sham Weizmann. He was actually British, and he discovered or discovered at least how to utilize this. So he would take Clostridium bacteria and was able to get them to produce all these things. And the British in World War I actually used acetone as a propellant, right? So a lot of times now, if you know anything about shooting, we use gunpowder, and the British were actually using acetone as a propulsion fuel for their artillery. All right, and these same byproducts we also see in the production or their usefulness in the production of different plastics, and so really, really important process for us. Now, Really quickly, this is not necessarily directly related to metabolism, but one of the things I wanted to point out is we focus really heavily on glucose, but some of you that have taken A&P also realize that as long as there are some type of polysaccharide or disaccharide present, some type of lipid or some type of protein, these can be broken down and then utilized to generate energy. So I don't expect you guys to memorize this necessarily. Well, these different pathways I'm about to show, but again, polysaccharides, right, complex sugars. We'll talk about some tests that look for the presence of enzymes that break down complex sugars. Not all bacteria have them. Not all bacteria have lipases or proteases that are breaking down either fats or proteins to utilize as energy sources. And this is just to give you an example, right? So here you have fat. When you start to break down fats, you have a couple different options, right? Fatty acids can be broken down via something called beta oxidation, and they can actually enter then the TCA cycle. So you can generate energy from them that way, right? You can also, when you break down fats, all fats are made of glycerol head and fatty acids. So that glycerol head can actually be used to make glucose, and then you could further break that down and use that as an energy source if you want. So again, I'm not expecting you to memorize this. I'm just showing you when I say, how can, you know, if you're asking, well, how can a fat be used as an energy source? How can a protein be used as an energy source, right? We can actually break them down and then enter them typically into the citric acid cycle. And then you can generate, right, your reducing power and your ATP there and get an energy. And this is just the same thing with protein. Right here, right, you break it down to amino acids, right? And the deanimated, right, you remove the nitrogen, and then from there, you can use that to help you generate different energy sources that you may need, okay? So cool alternatives to even organic fuel sources, right? So lipids, sugars, proteins, even nucleic acids are all organic energy sources, right? Remember, we've already mentioned some bacteria have the ability to utilize different energy sources. So I'm gonna skip these right here because they're summed up in this table. Basically what this is saying is that here you have hydrogen bacteria. So hydrogen gas is the energy source. Sulfur bacteria, so they can use sulfur right here. Some bacteria can actually utilize iron as an energy source. And you have nitrifying bacteria, which we'll briefly mention here in just a little bit, are taking nitrogen compounds, right? And using them for energy. And this is going to benefit us because then it will convert them to something that we can actually utilize after plants have gotten a hold of them. So again, just take a kind of look at this chart to just be familiar with some of those alternative inorganic energy sources, all right? And again, this kind of contributes to that idea of bacteria and even archaea bacteria being ubiquitous, right? I couldn't put you in an environment where there's primarily hydrogen gas, sulfur, iron, or even nitrogen and expect you to utilize energy and survive, but some bacteria can. All right. Stop the share there. What we're going to do here very briefly is talk about the Calvin cycle, and then I'll see where we're at, and we might talk about nitrogen fixation. So, wait, hold on. Share Calvin. Okay. Pull this bar. Sorry, guys. It's been. There we go. Let's put it here. Reduce that. Come on. That's gone. All right, there we go. Sorry about that. On the screen. Okay, so this is 10 slides. Carbon fixation and photosynthesis, right? You'll see why in just a second. I've kind of combined those, and it should make sense. So carbon fixation, you remember if you go back and you look at those charts, chemolithoautotrophs and photoautotrophs, primarily photoautotrophs, are going to be able to utilize CO2 to synthesize organic carbon from that, right? So basically you can take CO2 and then you're building those macromolecules that are necessary for life, This is why I linked it to photosynthesis. Carbon fixation requires a large amount of ATP. It's a huge investment. And we'll see here in just a second, a great place to get a lot of ATP from an abundant source is sunlight. Organisms that are capable of carbon fixation have an enzyme called ribulose biphosphate carboxylase, also called Rubisco, that allows them to fulfill this process. So even if your body decided, I want to utilize excess ATP to make organic carbon from inorganic carbon, it could not do it because we lack the enzyme Rubisco. Because this is such an important process in the world, Rubisco is actually the most abundant enzyme in the world. So you think about bacteria that are capable of this, you think about all the plants that are capable of this. This enzyme is essential to life. So it makes sense that it is abundant in large quantities. This is the process of carbon fixation. Again, I'm not going to ask you to remember this. I just want you to see that it requires a lot of ATP. So basically what you're doing is you're taking 6 molecules of carbon dioxide, and you are putting those carbons together to eventually get fructose 6-phosphate down here. It's this compound here, this fructose 6-phosphate, that is then the organic compound or the organic carbon that we know that we can use to build different things. These other compounds right over here are still important because they're allowing the continuation of the carbon cycle. Okay. So there you go, fructose 6-phosphate. The carbon cycle or the Calvin cycle is linked to both glycolysis and the pentose phosphate pathway, right? They can shuttle in important precursor metabolites that are needed to complete the Calvin cycle. Some of those precursor metabolites, right, are glyceraldehyde 3-phosphate, which can be generated in glycolysis or the pentose phosphate pathway, ribulose 5-phosphate, which is generated in the pentose phosphate pathway, and then NADPH is involved in carbon fixation in the Calvin cycle and is generated in the pentose phosphate pathway. So you can see, right, that the Calvin cycle is relying on 2, right, components of the central metabolic pathway. It's true you have the pentose phosphate pathway, but you're not able, right, to use these precursors like bacteria can. Again, 1 molecule of fructose 6-phosphate requires 6 turns of the cycle, right? That's the 6 molecules of CO2 that are coming in, right? A large ATP investment. And where does all this ATP come from? I've already revealed this to you, right? It's from photosynthesis. So the basic formula of photosynthesis, you take, right, inorganic carbon plus water. One of the multiple reasons it's important to water your plants, right? You have sunlight, all right? So you use that sunlight plus these molecules right here to generate, right, glucose, right, water, and in this case, right, you're going to generate oxygen. Right, so that's what the X is right here. Sorry, so you have 2 stages here. You have to capture the light, right, and convert it to radiant energy, right? So then you could utilize, right, this ATP in process number 2, and that's using the ATP to synthesize organic compounds. One of the big factors here, right, is that enzyme Rubisco. Is it or is it not present? All right, so here's where it gets a little fun, and I promise if you take the time to look over this, it won't be too bad here, all right? So what we're looking at here, right, is a photosystem, right, that's embedded within a chloroplast. So if you've done anything with plants, you've heard of chloroplast. Bacteria can have chloroplast equivalents if they're capable of photosynthesis. Also within, right, this photosystem, you have individual molecules of chlorophyll, right? That's where chloroplasts get their name. Chlorophyll capture radiant energy, all right? And once they capture that radiant energy, that's what's going to allow us to convert it into ATP. And so here you see when these chlorophyll capture this radiant energy, it ends up exciting an electron. And this electron is then going to enter the electron transport chain, just like we see in us and bacteria, and you'll see some differences there. Now, there are different pigments. So you have chlorophylls, you have bacterial chlorophylls, and you have other accessory pigments. These all allow you to capture different wavelengths of light. If you can capture different wavelengths of light, you can inhabit different environments. What we typically see with bacteria that are capable of photosynthesis is they are aquatic, all right? If you've ever swam underwater, if you've gone snorkeling, if you've gone diving, you'll notice, right, that things look a little different underwater. And one of the reasons they look different underwater, right, is that different wavelengths of light penetrate different depths. And so by having these different pigments, some prokaryotic cells can actually exist within different depths of water, which allows them to utilize those wavelengths and avoid competition, right? So here, these are just these different accessory pigments that I mentioned. So you find chlorophylls in plants, algae, and cyanobacteria. Bacteria chlorophylls are almost identical to chlorophyll. They're capturing different wavelengths. They're important in anaxionic bacteria, so bacteria that are not utilizing oxygen, but maybe in an aquatic environment. And then different pigments. So if you've heard of carotenoids or phycobilins, again, capturing wavelengths and allowing different environments. So here, this is just really similar to what we see going on in our bodies or bacteria that aren't capable of photosynthesis, but the same idea, right? So photophosphorylation, right? So we're capturing radiant energy, and again, bacteria to fuel ATP synthesis. Just like in us, right? In bacteria, you do have the generation of NADPH or NADH, right? And both of these are needed for carbon fixation, right? So if you're doing that, that's important here. So this is, okay, good. This is my last slide. So this is a little confusing, but if you really take the time to go through it, I think you'll understand, right? So again, some bacteria are capable of photosynthesis, so they have this going on. This is also what we see going on in plants, right? So you notice two photosystems here. Unfortunately, I'm sorry, I did not name these. They're kind of, if you want to say, if there's an order to this, they're out of order. That's because photosystem 1 was discovered first. We later discovered photosystem 2, which we typically start the process with. And so here, right, photosystem 2 is going to capture radiant energy, and it's going to capture it via that chlorophyll. This could also be bacteriophil, right, if we're talking about those anaxionic bacteria, all right? So chlorophyll captures radiant energy. It excites an electron, all right? And when those electrons get excited, they enter the electron transport chain. And what do you know? They are carrying protons with them. And so here you have a proton pump, the same basic principle to proton pumps that are found in eukaryotic and prokaryotic cells. It creates a gradient. This gradient will then be used to power ATP synthase, right? And so they'll make ATP. Now, the electron continues on, all right, to photosystem 1. And when radiant energy hits photosystem 1, that same electron can then be re-excited and enter the electron transport chain again. So this is a unique capability of photosynthetic organisms, bacteria or plants, that we don't possess, right? They are able to recycle that electron over and over and over again to generate energy, right? So radiant energy is incredibly good at generating ATP, and this is one of the reasons, all right? Now, what we also have happen, right, is some of these electrons are then used to generate NADPH. And this NADPH will then be used later for carbon fixation. If the electron goes this route, it can't be recycled. It can't be used again, right? It's hanging out with NADPH. But that's fine because photosystem 2 is constantly supplying us with electrons, all right? And so we can either keep on recycling them. If we use one up, no big deal, because we're getting more from photosystem 2. Now note down here, another unique function of organisms that are capable of photosynthesis, right, is this water splitting enzyme, right? So in this case, the electrons that these organisms that are capable of photosynthesis are using are coming from water, right? This is why one of the reasons that water is super important for plants. It's their electron source for the electron transport chain. Okay? So I feel confident if you take a look at that, it'll all kind of hopefully sync together. Please don't ignore it. I'm not going to give you a 15-point photosynthesis question on the next exam, but there might be a 1 to 5 point question potentially related to it. So please look over that. I know I'm going through kind of quickly. I kind of want to get a little bit back on track. So please look over this and please look through your textbook and ask questions if you have them. All right. The last thing that we'll talk about that should not take up too much time, hopefully, is the nitrogen cycle. And this has 8 slides, so this will be really quick for us. And, truth be told, you could spend, a whole unit, or maybe even a whole semester on these different cycles, but that's just not the amount of time that we have. So I at least want to kind of go over it briefly. All right. So nitrogen, right, exists in abundance as nitrogen gas, right, an important part of our atmosphere. But nitrogen as a gas, N2, cannot be utilized by organisms like us, right? Those chemoheterotrophs. But, right, what can happen is nitrogen can be reduced to ammonia, and ammonia can be incorporated into plant cells, right? And plant cells can then turn that ammonia into organic nitrogen compounds. And when we eat them, right, we can take that nitrogen because it's then in a form that we can use, or we eat things that eat plants, and that's where our nitrogen's coming from, right? An enzyme that's needed to do this is an enzyme called nitrogenase, and it requires a lot of energy because it's breaking down nitrogen gas, which has a triple bond. I know some of you run from the hills when it comes to chemistry. Maybe you just have blocked this out of your mind. But remember, when we have covalent bonds, you can have or kind of, some common things are single bonds and double bonds. Every time, right, you have an extra bond there, that means you're sharing more electrons. It means it's a stronger connection. So nitrogen gas actually has a triple bond, which means it is a very strong molecule, requires a lot of energy to break it up. Groups of prokaryotic cells called diazotrophs are the only organism, right, that can fix nitrogen. And there's different groups within this group, right? But diazotrophs are prokaryotic cells that are capable of breaking nitrogen gas down into, right, compounds that can be utilized by plants. And so you have a couple different options here. You do have some free living ones, Azobacter, Clostridium, and Berginkia, right? Oof, I don't make some of these names, right? They can just do this. They're out on their own. You can even grow some of these in culture. The ones that we become really interested in are those that are associated with root nodules, right? So Rhizobium, Frankia, and Azospirillum, right? So these associate with plants. They break down nitrogen. Plants then take up, right, that nitrogen in the form of ammonia and can incorporate it into their organic nitrogen compounds. Here you just see a couple different processes that are occurring. So nitrification, right, denitrification, right? There's a bunch of different things going on here. One of the ones that I really want you to focus on, though, here, right, is nitrogen fixation. So you're taking nitrogen gas and you're converting it into ammonium, right? Ammonium, you can see here, right, can then be incorporated as organic nitrogen or turned into organic nitrogen, where it can be utilized by plants. You see there's different options here, right? Bacteria can also take nitrate, right, that was generated early in the process and through something called nitrate assimilation, right, can turn it into organic nitrogen, which can then be utilized. And so the big thing here, right, nitrogen fixation makes ammonium. Ammonium can then be incorporated into organic compounds. And this is what we see going on, right, in the relationship between those bacteria and plant nodules, right? So this is kind of the most important one. Here, I go through these different processes so you can kind of, look at them. But if you go back at that cycle, you look at ammonification. Here you're seeing organic nitrogen converted into ammonia. And again, this can be utilized further by microbes or even by plants. And so there's a lot of different things going on here. Again, I'm not going to ask a big question. If you're more interested in nitrogen fixation, these are kind of some simple videos that kind of go through that process for you. What I'm really trying to get at here is that, right, we need bacteria that are associated specifically with root nodules that are taking that atmospheric nitrogen and converting it into a form of nitrogen that can be utilized by plants, and then we, kind of go from there. So it is 10:10. What I'm going to do, if it's okay with you guys, is I'm going to say, let's just go straight into lab. I'll break you up into your groups in just a second. What I wanted to do, so what you guys are going to do for lab today is at least give you a brief introduction of kind of what you're looking at. So if we were in lab, right, we'd be utilizing a basic light microscope to identify different bacteria via stains and then morphological differences, right? So what is their shape? How are they clustered together? Sometimes they'll exist as chains. Sometimes they'll exist as kind of individual organisms that are spread out. Sometimes they'll cluster in pairs. Sometimes they'll form clusters that look like grapes, right? And so microscopy is very important for that. And so your kind of two lab activities today are looking at the basic components of a microscope. What are they doing? You can use the internet as you go through these. There's also a microscope chapter in your textbook that these questions and these activities have been pulled from that may be useful. The other thing you're going to start to look at is sample preparation. So how do you prepare bacteria, or how do you take bacteria from a liquid culture or a plate and put it on a slide to get it ready to stain? All right. It unfortunately is not as simple as just taking a glob of bacteria, putting it on a slide, and then being ready to stain. You'll see that you go through one of the most important processes is heat fixation that will be covered in here, right? And you're basically lightly cooking the bacteria to the slide so that when you stain it, it doesn't wash off, right? And then kind of what we'll do for two other labs this week is you'll go over one of the most basic important stains, which you guys have encountered a little bit already, the Gram stain, right? You will then go over a couple other different stains, specifically the acid fast and the endospore stain on the days following that, right? On Thursday this week, what you guys will be working on together in your lab groups are basically small little, I kind of call them mini presentations, where each group will be assigned a couple of different tests that we would have done in lab. And you'll see this packet. I'm going to post it on Moodle probably today, and I will send out an email with the groups and then the tests that you're responsible for looking up. And there's, I think, a little bit of a description about this in the week 2 schedule. But the idea would be you look up the basic information of the test. What is it testing for? How does it test for that bacteria? And then what are bacteria that commonly are identified by that test? If we were in lab, this would be a packet that I'd give you early on, and you'd keep it with your lab notebooks. So as we went through and actually did these tests, you would be able to refer back to this packet and kind of figure out what they're for. And then as we move forward, right, next week, some of the lab activities and assessments will actually be kind of part of this, or will be some of the ones that you go over. The idea is, right, your group looks at like 4 or 5 of them. You present that, or you share that. We're going to share that all together, that information, on Monday. So then you guys have a basic understanding or idea of what some of these different tests are doing. I think I put this in the description. What I would like for you guys to do is search on the internet and find an example of what a positive test looks like and what a negative test looks like. All right. But again, this shouldn't require a ton of work outside of class time, because I'm going to give it to you to do. That's going to be what we're doing in lab on Thursday. In lab on Thursday, and you guys can work on it as a group. And as soon as I send it out, you guys can start working on it then if you want. On Thursday, I'll kind of show you an example of what I'm kind of expecting. And I promise this will be beneficial, because again, you're going to be filling out information on some of these tests and your mastering assignments next week. This will also kind of start to set up your unknown, right, which is your big lab project. And so this is important for that and helps you with that. Okay. So I'm going to break you up into groups. You're going to do the microscope and the slide preparation mastering assignments today, and I will work on getting those group assignments and the tests that you're supposed to work on out to you today. But again, we'll work on those during lab time on Thursday. So if you can't get to it, if you don't want to think about it between now and then, that's totally fine. I think Thursday will give you guys plenty of time to be working on that. All right. So let me divide you up into your groups. You can get started on those mastering assignments, and I'll pop in and out as usual. Right.